Thank you for the opportunity to share the story that I've been working on for the past 2 years. And this paper is ready, but I don't have time to show you all the data. So please come talk to me after, and happy to collaborate. So we began by, I'm preaching to the choir here when I say, um, desmoid tumors are clonally proliferative, but locally infiltrative, associated with beta-catenin and APC mutations. But our hypothesis when we began the study was there are several key pathways enriched in DTF patients. And while we knew that beta-catenin is a major player, we also found that c-Jun, which was the protein that we were interested in, is a key player in the pathogenesis of DTF. So we found that there were several hallmark pathways, which are the EMT pathways, extracellular matrix pathways, and we wanted to identify if we could find some immune targets that were driven by c-Jun, which could be therapeutically targeted for desmoid tumors and other sarcomas. So we began by looking at bulk RNA sequencing data to see whether we do have pathways that are enriched specifically in the desmoid tumors relative to normal controls, and what are these pathways associated with. And so we found that ECM pathways and the collagen formation pathways, which are profibrotic pathways, are enriched. That was kind of obvious. The next question we wanted to know was, within these pathways, do we have a master regulator that is driving these genes? And so we found that c-Jun was indeed a regulator driving all the genes that were driving this profibrotic pathways. And so we knew that this is a transcription factor that regulates cell proliferation and survival. But our question was, what is a downstream target of c-Jun that we could potentially use as a therapeutic strategy? So we began by saying, can we understand the cell type profile and gene expression patterns in desmoid tumors? And so I really like to combine omics approaches to ask, to delve into these questions. And so we built this TMA with 35 patient samples and 5 controls. And most of the samples here were, fibrotic cells and several tumor infiltrating immune cells. And I performed cosmic spatial multiomics on these samples and combined it with single cell RNA sequencing. And so now we have information that is on the spatial level in a patient cohort that is more than 30 patients. And we did this at the transcript level as well as at the protein level. So right now the technology is only developed for 960 targets at the transcript level, and at the protein level it's 64 targets. And I've done both and combined them and combined this data with single cell RNA-seq and bulk RNA-seq. So we have a pretty, deep understanding. So these are to give you an example of the TMAs that we built. And so what we found was that desmoid tumors display distinct cell clusters. And I'd never seen people look at the macrophage population within these tumors. And so while we focus a lot on the fibroblasts, we've not taken into account the immune component of these tumors. And so we looked at these distinct clusters and okay, we looked at these distinct clusters, and then we found that there were 5 different cell types that were distinctly expressed between the tumors and the normal tissue. And we found that there was this very characteristic fibroblast cluster that was displaying the ECM phenotype that had EpCAM. So you see, and this was a cluster that we were confused. We're like, well, it's epithelial, but it's also fibrotic, and what is it? And so we found that in our desmoid tumors, there were these ECM phenotype, cell clusters that no one in the world had looked at before. And then we focused on the PDGFRA positive, and this is combining spatial data and single cell RNA-seq data. So we validated it at both levels. And when we focused on these PDGFR positive cell populations and looked at, so you can see the ones that I've highlighted are the clusters that are Jun positive enriched in the patients. We found that while there were these enriched pathogenic fibroblast clusters, there were also distinct macrophage populations that were enriched in these tumors. So you can see we use CD68 to cluster all the macrophages, and then we focused on the M1 and M2 phenotype of macrophages between the, the inflammatory, pro-inflammatory and anti-inflammatory phenotype. And what we found was that the M1 and M2 phenotype of macrophages were differentially expressed between normal and patients. And I know, I think we've not looked at this before, and it's something we need to consider, the immune component along with the pathogenic fibroblasts. That when we look at patients, we see that these M2 are higher compared to the M1, which we see in our normal donors. So we found that these macrophages, the very specific, M2 macrophages, are enriched in desmoid tumors, which we don't find in our normal tissue. And so we wanted to characterize this further. But going back to the question, so now we've looked at pathogenic fibroblasts as well as macrophages. So what do we find? Do we have an immune target that we see in the pathogenic fibroblasts? And we found this target using multiomics. So we did Codex, we did Cosmics, Codex, single-cell RNA-seq, and CyTOF to combine and find this target. And we found that CD63 was one of the targets that was enriched in our desmoid tumors. And it's very specific in the pathogenic fibroblast populations, which you don't see in the normal donors. And this is from the single-cell data with spatial. So what is 63? CD63 is regulated by Jun. It has not been shown before, but I have another paper that shows that Jun regulates 63, and it's a tetraspanin. Then I wanted to look into the single cell. So these are the primary cell lines that we got, OSU DES23 and RCF, which we got from Jesse Boehm's group. And we found that CD63 along with CJUN are indeed overexpressed in the patient samples compared to the controls. And so then I overexpressed Jun and knocked out Jun and showed that there is a change in the cell proliferation rate in these primary desmoid cell lines. And I found that this was the same case when I overexpressed and knocked out CD63. And we found that Jun specifically transcriptionally regulates 63 in these primary desmoid human cells. Then we did ATAC-seq, and then I did single-cell ATAC-seq to look at just the pathogenic fibroblasts within the desmoid tumors. And we found that if you look at the promoter regions here, that Jun is binding to the promoter of CD63 and driving it. So we have a mechanism that explains how CD63 is driven by CJUN, and CD63 promotes desmoid tumor progression and works in concert with the immune environment. So it's the pathogenic fibroblast with the macrophages. And I confirmed this with CHIP to show that that Jun is binding to the promoter of 63 and driving it. Then I knocked out Jun and 63 to show that the profibrotic gene signatures were reversed in primary desmoid cells. And this is, again, we show genetic knockout of Jun and 63 to reverse the profibrotic genes, which are FSP1, collagen A1, that get reversed when you knock out Jun. And when you overexpress Jun and you knock out 63 and you overexpress 63, telling us that CD63 plays an important role in the progression of desmoid tumors. So the question is, can we safely target 63 for DTF treatment? So we made a monoclonal antibody in the lab. This is how we generated. We used hybridoma technology, and we made the CD63 anti-human targeting CD63 antibody. And we show that when you block 63, it leads to the desmoid cells get cleared by a recruitment of macrophages through phagocytosis. So you can see here with the CD63 knockout, there is a significant increase in phagocytosis. So our mechanism of action for this drug that we're trying to, it's right now in preclinical, testing. We think that it works through phagocytosis, so it elicits an innate immune response. And we now have a downstream target for CJUN that we can potentially target as an immune checkpoint therapy in combination for desmoid treatment. So this is our strategy as a treatment strategy. So in summary, I think we've performed multiomic profiling. So we have spatial multiomic data and single cell RNA-seq data and bulk-seq data, and we've combined all of this to understand the different cell clusters that are present in desmoid patient tissue. We've found CD63 as an immune protein that is enriched within the pathogenic fibroblast clusters. We know the mechanism through which 63 is regulated, that is the AP1-C-gene nexus, and overexpression of C-gene drives this diseased phenotype. And we also have a treatment strategy that we want to, potentially bring to clinic, which would, combine targeting the pathogenic fibroblast and eliciting an immune response. So yeah, that's just a gist of the story. The paper is ready, so please reach out. We have all the data. Please don't repeat this, the spatial transcriptomics again, because we have it. And so we're happy to share, and we have the TMAs ready as well. This is the team. I'd like to acknowledge Galinda, who is my mentor. I'm now an instructor. Desmoid Tumor Foundation for funding my research and giving me the postdoc fellowship, and the Broad Institute, Jesse's group that has shared cell lines with us, and OSU, of course, that sent the Pollock group that sent all their samples to Stanford. It was a huge process with the MTA. Yeah, for this collaborative environment, which helped us identify an immune target and dissect the cell clusters, both spatially and quantitatively. So now we not only have an understanding of the cell clusters, we have a target and we have a drug. So yeah, I have an ask. I'm interviewing for faculty positions. So yeah, in Jan of 25, I want to start interviewing. So I thought I'd share my plans. I'm interested in collaborations. I'm in Europe a lot as well as in the US. So if anyone wants to collaborate, has patient samples, we have the technology at Stanford and I have the expertise. So we could have joint projects, and I love to give seminars and present my work. So if any of you are interested in working together, that's my email. And yeah, you can find me on Stanford's website, and happy to talk further, share data and everything. Let's make this happen.
And so we're open for questions. Anyone have questions?
Jesse.
Jesse, go ahead.
First of all, I want to comment that if I had any say over my hiring at my institution, then I'd hire you in a second. So,
Oh, yes, please, MIT, take me.
Created so valuable, very foundational, like lots of LC. Want to do new algorithms to try to find interactions between the cancer cells. So, where do you think this data can be hosted so that the most members of this community can analyze it in a number of ways?
Yeah, so that's what, and I was going to reach out to you. I think you're building this, yeah, because you're building the atlas, right? And I think it would fit really well because we can spatially see the cells, and we also have the quantitative data. So I do think if there's some effort that we can make to put this data together in on one, have a repository where we can combine. We have the single cell, we have everything, right? So it's the multi-omics, we can put it out there so people can look at additional targets as well, and also the different cell types in patients, which is very specific. Also, different patients, you see different cell clusters. Yeah.